Welcome back to this next video in which we are discussing the human papillomavirus entry mechanism. Okay, so let's just summarize where we've got to in the story so far. So, the human papillomavirus particle enters this area where we have had the microabrasion, and the first thing that has to happen is it has to bind to the primary receptor, and the primary receptor is heparan sulfate proteoglycans. The L1 proteins, the major capsid protein, can bind to heparan sulfate chains. Now, there are heparan sulfate proteoglycans, as we've discussed, in both the basement membrane and on the surface membrane of the basal cells. However, we think that probably the virus initially binds to heparan sulfate proteoglycans in the basement membrane just because of the higher probability that it will come into the microabrasion site and hit the basement membrane first. So we think it probably initially binds to heparan sulfate proteoglycans in the basement membrane, and then as the healing process begins, as the basal cells start to regenerate the layer, you'll get basal cells moving over this surface where the virus is, and therefore they'll be exposed to the heparan sulfate proteoglycans on this basal portion of the basal cell membrane, and it will then be transferred onto the cell membrane. So let me just draw a little picture of this. So moving on to the next picture, I'll just put a little arrow here. So here, this is the basal membrane of the basal cell. So effectively I'm just drawing the same picture as I had previously. This is another basal cell that's now moved over, we're imagining, this area here. And of course, in its membrane it will have heparan sulfate proteoglycans, and I'll try and draw it similar to how I've shown it in the other picture there. So here is the core protein, the syndecan 1, we think is particularly important, and then of course coming off it you'll have lots of heparan sulfate chains. So here in green are the heparan sulfate chains, and now the virus particle is now going to be bound onto those heparan sulfate chains. So here we go, here is the capsid in red here, and then the viral genome in blue. Okay, so this is the stage that we've got to at the moment. The virus particle is bound to heparan sulfate proteoglycans on the surface of a basal cell. How is it now going to get inside of that basal cell? Well, you might think, okay, endocytosis is going to follow. Now it's bound to a cell surface receptor. All that needs to happen now is this needs to be endocytosed and take the virus particle with it. However, we do not think that the heparan sulfate proteoglycans on the surface of the cell membrane are the actual entry receptor for the virus. We don't think this is how the virus is going to be endocytosed, and it is going to be endocytosed, but not through this receptor, we don't think. Therefore, we refer to the heparan sulfate proteoglycans as the primary receptor or the attachment receptor. They are not the entry receptor. So this is the primary receptor, meaning the first receptor that the virus particle binds to, but there's going to be a secondary receptor, and we don't really know the identity of that secondary receptor, but we'll cross that bridge when we get there. So this is the primary receptor, also called the attachment receptor, and this is a nice name for it because this tells you that really all this is for is the attachment of the virus to the surface of the cell. It's not going to be the way that the virus actually enters the cell. It's not the entry receptor. That's going to be the secondary receptor. Okay, so how then is the virus now going to be transferred to a secondary receptor? We might think now it's on the surface of the cell membrane, maybe it's just going to be transferred to a secondary receptor. However, we think that in order for the virus to be transferred to a secondary receptor, you have to get conformational changes in a lot of the L1 proteins to allow them to then bind to the secondary receptor, whatever it may be. And where do these conformational changes come from? Well, we believe that they are facilitated by an enzyme that is also bound to heparan sulfate proteoglycans on the surface of the basal cell membrane, known as Cyclophilin B. So this is the next player in our story. So next up, we're going to discuss this enzyme called Cyclophilin B. And for short, cyclophilin B is often abbreviated down to CYP, specifically like that, where you've got a capital C, a lowercase y, and then a capital P. Do not confuse this for the cytochrome P450 enzymes, that are really important in the liver. So CYP like that means cyclophilin, and then we're talking about cyclophilin B, so CYPB, like so. So cyclophilin B is an example of an enzyme known as a peptidyl prolyl isomerase, and I just want to spend a little bit of time explaining what is actually meant by this term. So this enzyme is an example of what is called a peptidyl prolyl isomerase, and it's going to catalyze the change in conformation between a peptidyl prolyl bond in a polypeptide. So peptidyl prolyl isomerase, it's going to move you between two different isomers of the molecule, two different conformations of the molecule, so it's going to facilitate conformational changes. So let me just explain what is meant by a peptidyl prolyl isomerase in detail. So this will require me to draw a bit more biochemistry, so back to biochemistry we go. So I'm just going to draw a peptide bond. So I'm going to draw two amino acids in a polypeptide bound to one another. So we'll start off here. So here's the first amino acid. Here's its amino group, and I'm again sticking to skeletal structures. So this would be where the amino acid in front of it's linked by a peptide link, but I'm not going to bother drawing that amino acid. Then we have the alpha carbon here with some R group, which I'm just labeling up as R1 here. Then we come down to here, we have the carbonyl group here, which will be amide linked to the next amino acid along. This is the peptide link that we've got here. So this is one amino acid residue in the polypeptide, and here is the next amino acid along. So here is its alpha carbon, here's its R group, which I'll just call R2 here, and then here is its carbonyl group up here, like so, and then it will be linked to the next amino acid along. So I've drawn you two amino acids here. Here's the second one, here's the first one. Now, this is one conformation that this peptide bond at the centre here can actually be in, and this is what we would call the trans conformation of the peptide bond. Now, trans means on the opposite side of, and the reason this is called the trans, the on the opposite side of conformation for this peptide link, is if you look at the two members of the peptide bond, this carbon here and this nitrogen here, the major groups, which are the continuation of the polypeptide chain, of this carbon, this continuation goes off this way, whereas off the nitrogen, the continuation of the polypeptide chain goes off the opposite way, so they're on the opposite sides, you see. This hydrogen and this carbonyl group are the minor groups, these are the major groups. So we would say that the two major groups, which are the continuations of the polypeptide on either side of this peptide bond, are on opposite sides of the peptide bond, and therefore this is the trans conformation. Now, in theory, you can just rotate this bond around and you can get it into the other conformation. So let me now show you the other conformation. So what I'm going to do is I'm going to keep this one in the same position, and I'm going to rotate this one around by 180 degrees. I'm going to bring this bond here into the position that this one's in, and this one's going to be spun up there. So I'm just going to draw a little arrow for what I'm going to do. I'm going to spin it round like that into a different conformation, which in theory you can do. This bond is rotatable. It can rotate. So let's do this. So I'll go back to the pink pen. So here is this nitrogen. I'll get my laser pointer. It's this nitrogen here. And here is the hydrogen coming off that nitrogen. Here is its alpha carbon here, its R group down here, R2, and then the carbonyl group up here, and then that will be linked to the next amino acid along. Now, here's the peptide link again, but this time the carbonyl group is going to, well, the double bond to the oxygen is going to come off this way, like so, whereas the other group, the major group, is now going to go off in the opposite direction. So here comes my visualization ability. How good is it? if we rotated this around 180 degrees, this one would then point down here, I think, and this one would then have to come off over here. So like so. So here is the R1 group, and then here will be our nitrogen, and then which direction will the hydrogen end up pointing? Ooh, would I like to make a guess of that? So think about this. Here is this bond here, so I think the hydrogen would end up having to come off down there, like so, I think. And then the other group would come off like that. I think that's what it would end up looking like. Okay, so in principle, you can rotate this molecule around and have it in this conformation like so, and this we would call the cis conformation. Cis means on the same side. So trans on the opposite side, cis on the same side, and now the two major groups are on the same side of the peptide bond. Now, in reality, peptide bonds never exist like this. They do not like existing like this. Well, I shouldn't say never. Never say never. But they don't like existing in this form generally. So most peptide bonds will not exist in this form. They'll be in this conformation. This is the preferred conformation for a peptide bond. However, if this second amino acid was the amino acid proline, then that rule that I've just given you, that they don't like being in this form, that is, no longer applies. If it's proline, then actually it can exist in both forms, and therefore the polypeptide has these two possible conformations at that peptide link, and these enzymes are going to switch them between the two conformations. Now, just to remind you then of the structure of proline, because that will be one of the major motivating factors for why proline is different. Remember, proline is a very different amino acid from all the other amino acids. So we're now going to talk about this amino acid, proline. Okay, so let me draw one more picture then of polypeptides. So I'll get another colour to make it more interesting. So I'll have red now. So the first amino acid is just going to be a normal amino acid, i.e. not a proline amino acid. So here it is. Here's its amino group. Here's its alpha carbon. Here is its R group, R1 up there. Here's its carbonyl group down here. And then the second amino acid is then going to be proline, and this is why these enzymes are called peptidyl prolyl. It's referring to the peptide chain, the normal peptide chain, and then the proline. It's referring to this peptide link between the peptidyl chain in front and then the proline. So now let me put proline in here. So here is the nitrogen, here's the alpha carbon, here is the carbonyl group here, and remember the R group of proline is weird because it involves the amino group of the amino acid, whereas no other amino acid that's used in proteins normally involves the amino group. They are like the pictures that I've drawn here where the R group just comes off the alpha carbon, it doesn't involve the amino group at all, whereas the R group of proline consists of 3 carbons forming a ring like so between the alpha carbon, a 5-membered ring involving the alpha carbon here and the nitrogen here. So this is a corner, this is a corner, this is a corner. We have 3 carbons there forming a ring, and each of these carbons has 2 hydrogens linked off to it. So this is the R group of proline. So it's a very funny amino acid, and because of this, the peptide bond here can exist in these 2 conformations, trans and cis. Of course, this is the trans one. You can imagine then the cis one here. I'm not going to draw it out, but of course, just rotate this round again 180 degrees, just as I did here. So when we have a peptide bond involving a proline, so a peptidyl prolyl bond, and I'll just write this here. So we have a peptidyl prolyl bond here. It can actually exist in the 2 conformations, and it can exist stably in the cis conformation, whereas other peptide bonds, they cannot exist stably in this form, and they just instantly turn back to this form, whereas peptidyl prolyl links can actually stay in the cis conformation. And these enzymes then, quite simply, they twist the protein around. They change these peptidyl prolyl links from the cis to the trans, or from the trans to the cis. They move them in between these 2 conformations. and therefore they are fantastic at changing the conformation of the enzyme. By the way, for short, peptidyl prolyl isomerases are often abbreviated down to PPIases for obvious reasons. Okay, so now we know what cyclophilin B is. It is a peptidyl prolyl isomerase. It has the ability to change the conformation of proteins by changing the conformation of these peptidyl prolyl links. Now, cyclophilin B is present all over the place inside cells, but also we think it's actually present bound to the outer surface of cells. Specifically, we think it's bound to heparan sulfate proteoglycans. So let me just draw this on here. So here we have my little enzyme drawn here in yellow in the classical way that enzymes are drawn with the active site there. And this is representing cyclophilin B bound to the heparan sulfate proteoglycan. So isn't this a wonderful thing? We have bound the virus to heparan sulfate proteoglycans on the surface of the cell membrane, and now they're brought directly into contact with cyclophilin B enzymes. And we think that cyclophilin B enzymes are responsible for helping in conformational changes in both the L1 and the L2 proteins. So, the next part of this story then is that L1 and L2 proteins in the capsid are going to undergo conformational changes facilitated or catalyzed by the enzyme cyclophilin B. So I'll just put this here, L1, L2, conformational change. And this conformational change in L1 is what's going to make it then able to bind to the secondary receptor. So it's going to enable it to then bind to the receptor that's going to allow you to actually enter cells. Now we'll come on to that again later. So conformational changes in L1 and L2. For now, I want to actually talk about the other one, the L2 conformational changes. So conformational changes in L2. Now remember, L2 is not essential at all for viral entry up until right at the end. So if you have a capsid that doesn't have any L2 proteins at all and just L1 proteins, it gets very much so along the pathway. It's just it doesn't quite get through the endocytic pathway in the same way without L2, but it certainly still binds to the heparan sulfate proteoglycans, and it still gets transferred to the secondary receptor, and it still gets endocytosed and ends up in the endocytic pathways. It's just it doesn't escape from the endocytic pathways in the correct way. So L2 isn't going to be, important right until the end. However, despite what I've just said, I'm now going to tell you all about what the L2 conformational change is now going to do, because in the time phase of the entry mechanism, L2 now undergoes some important change that's going to be important later on, but it's not going to be important imminently. So I'm going to tell you now about what's going to happen here. But it's not going to be important until later on. It happens now, but it's not going to be important until later on. So it's the L1 conformational change that's going to allow us to bind to the secondary receptor, not what I'm about to tell you about L2. So, with that warning then, what happens to L2 now? So, cyclophilin B is helping to catalyze conformational changes in loads of L1 proteins and L2 proteins in the capsid. When L2 undergoes this conformational change catalyzed by cyclophilin B, it exposes a little bit of its N-terminus. It exposes its amino terminus. So let me just remind you of how L2 sits in the L1 pentamers. So just drawing a little picture here of an L1 pentamer, like so. So remember, the capsid is created out of loads of L1 pentamers, also called L1 capsomeres. And I'm just wondering, did I give you that piece of terminology? It was so long ago since I made the video on the structure. I'd just like to, I'm just doubting myself whether I gave you that piece of terminology, so I'm just writing it down again. So remember, a name for an L1 pentamer, another name for it, is to call it a capsomere. So it's just another name for an L1 pentamer. So we can say that the capsid is made up of 72 L1 capsomeres, which are the L1 pentamers. I don't know if I did give you that piece of terminology earlier on. I apologise if I didn't, but you've got it now anyway. Okay, so here is an L1 capsomere then, a pentamer of L1. 1, 2, 3, 4, 5 L1 proteins, and then remember it has that central tunnel which the L2 protein can sit in. So here is the L2 protein, and then it will have a domain inside the capsid down here. So it has a tiny bit that's exposed to the outer world, and which is this bit up here. Okay, now the cyclophilin B, when it facilitates conformational changes in the L2 protein, is going to cause a conformational change up here, and what's going to actually happen is a portion of the amino terminus of the L2 protein is going to be exposed like so. And then what actually happens is that amino terminus is cleaved off. The first 12 amino acids are cleaved off. So 12 amino acids are going to be cleaved off when the L2 protein undergoes this conformational change. And that cleavage is going to be done by enzymes that are in the extracellular space. And there are two enzymes that we think are important. One is called furin, and the other is called proprotein convertase 5/6. And both of these are endoproteases. They cut proteins not from the outside but from the inside. So proprotein convertase 5/6, and proprotein convertase 5/6 is just one enzyme. I don't know what the story behind it being called proprotein convertase 5/6 is. I imagine what happened is someone named it proprotein convertase 5, someone else named it proprotein convertase 6, potentially in different animals, and then it was acknowledged that actually they were the same enzyme, so it then became known as proprotein convertase 5/6. Often it's just, abbreviated down to PC for proprotein and convertase 5/6, like so. So as I say, both of these enzymes, furin and proprotein convertase, they are examples of endoproteases that are present in the extracellular space. So these are endoproteases. So remember, a protease is something that's capable of cleaving peptide links. An endoprotease is one that cuts peptide links in the middle of proteins. So if this is the polypeptide, endoproteases can cut them right in the middle, whereas ectoproteases, they can only cut the first amino acid off, then they can cut the next one along, so they can only cut the amino acids that are right at the end of the protein off. They can't cut peptide bonds right in the middle of polypeptides, whereas endoproteases can. So both furin and proprotein convertase, 5/6 are both endoproteases in the extracellular fluid, and when cyclophilin B causes this conformational change in L2 that exposes its amino terminus, they cleave off the first 12 amino acids. So this fragment goes off, and then L2 is left without those first 12 amino acids. Now this is not important until much later. It will become important for L2 functioning when it's going to function, but this is not important for the virus particle now being transferred to the secondary receptor, but it does happen now. So continuing on the story, this is stage 4, so L2 cleavage by furin and proprotein convertase 5/6. That occurs next, but it's not significant until much later on. So now, what's going to happen now? I will tell you much later on when L2 becomes important, and the fact that if this doesn't happen, if this cleavage doesn't happen, if you inhibit these enzymes, furin and proprotein convertase 5/6, this cleavage doesn't occur, and then the virus gets stuck much later on in the entry mechanism when it's trying to get out of the endocytic pathway, because the L2 isn't functional unless it's had 12 amino acids at the start cleaved off. So this is going to become important. It happens at this point, but it's not imminently important. So, what's going to happen now? The virus is going to be transferred onto some sort of secondary receptor that is going to mediate its endocytosis. So let's have some interesting colour. Let's go for purple here, and we'll draw another picture, I think, now. So, we need to draw another basal cell then. So again, here is our basal cell membrane here. And at the moment, the virus is bound to heparan sulfate proteoglycans. So once again, I'll just draw one of these heparan sulfate proteoglycans on the surface here. So this is again representing syndicin 1 or some other cell surface heparan sulfate proteoglycan. So here are the heparan sulfate chains, and then we'll have the virus. So here is the viral capsid that has now undergone conformational changes. So L1 proteins, lots of them have undergone conformational changes. Now, it might not have been all of them. The cyclophilin might not have got to all 360 L1 proteins, but a lot of them have now had conformational changes facilitated by cyclophilin. In addition, you've had conformational changes in some of the L2 proteins, maybe not all of them again, but some of them, and that's then resulted in the exposure of their amino termini, and then they've been cleaved off by furin or proprotein convertase 5/6. And you don't need both of these. One of them performs the cut, and once that's done, the other can't come and do it again. It's just that both of them can perform this same maneuver, but you don't need both of them. Okay, just one is enough for each of the L2 proteins. Okay, so what's now going to happen, because of the conformational changes in L1, not because of the conformational changes in L2 and because of the cleavage of L2, because of the conformational changes in L1, this can now bind to a secondary receptor, and it will prefer to be bound to the secondary receptor than the heparan sulfate proteoglycans now. So here is the secondary receptor. So I'm doing it in this fancy purple colour to make up for the fact that we don't actually know what this secondary receptor is. So this is the secondary receptor, the one that it binds to second, and it has another name. Because this is the one that mediates the endocytosis of the virus particles, we can also call this the entry receptor. So heparan sulfate proteoglycans are the attachment receptor, or the primary receptor. This is the secondary receptor, or we can call it the entry receptor. And I'm afraid I'm going to have to put a question mark there. We don't know what the identity of this secondary receptor is at present, but what we do believe is that it exists. It is important in the entry mechanism. We do think that there is a secondary receptor other than the heparan sulfate proteoglycans, which the virus particle is going to be transferred onto, so it's now going to bind to this, and then what is going to follow is endocytosis of the receptor and the virus bound to it. So endocytosis will follow the virus binding to this receptor. Okay, so it will be taken up into an endocytic vesicle, and then it will begin its way through the endocytic pathways. Okay, so continuing on with our numbered sequence. So number 5 is transfer to the secondary receptor. So transfer to, and I'll just put second receptor. And then what's going to happen is endocytosis. So I'll pick another colour. We'll have red now. And the next thing that's going to happen is that the secondary receptor with the virus bound to it is going to be endocytosed. Now, we will have a break here, and in the next video we will study endocytosis. We'll study the different mechanisms of endocytosis, because I'm afraid it doesn't seem as though there's just one mechanism by which HPV bound to the secondary receptor can be endocytosed. It seems that for different HPVs, the endocytosis mechanism is different. So that potentially means that the secondary receptor is different for different HPVs. We don't know at present. But it is a very real possibility that this secondary receptor is not the same protein for all the different types of HPV, whereas the attachment receptor, we believe, is the same for all the different types of HPV. It might turn out that it's simpler, and that for all the different types of HPV there is just one secondary receptor, but it may turn out more complicated and there are many different secondary receptors. However, for all of them, what has to happen is this is transferred to that secondary receptor, and then endocytosis is going to follow. And I'm just going to make my no, I can't. I'm going to try and get rid of that. Whoops. No, it's not going to work. Okay, we will have a break here, and I'll see you in the next video.